Hello, sir.
Yes.
Sir, but in that question, all of these was also there as an option. So is it wrong to mark all of these?
Which one?
Sir, in the question you mentioned
Now, all
actually.
of this is not the right answer because if you see, right, I'm just opening the question one minute.
The option C was all wrong.
Yeah. The extra carbon atom, you cannot answer that. Even I would say, even option A is not that way, right? Because in Google, if you type, you will get many like many like option or answer. But in any standard textbook, if you just go through it, or say for example, if you just score it that 100%, I would say 99%, like the primary reason is just having to prime age group. Even in your slide, you will get it. Any standard textbook, you will get it. And option 3 is wrong definitely. So all of the above is not the right answer. But I understand that that some confusion between option A and B. That means thymine uracil as well as here to prime OH. But that might happen. That's why I'm discussing this question, right? So that is okay, no problem. So like after like few classes, if I get more time, then I will go through all this assignment. Like you just got one assignment, and then next week you will get another one, and at the end I will discuss all those assignment also. Okay, but this question I have seen that many of you have like little bit of confusion. That's why I just discuss.
Okay, thank you, sir.
Thank you. So just little bit of just we have to remember what we were discussing last time. So now we are talking about transcription. That means uh, whatever DNA present in our body, in our cell, then from that information, how much work can be done. That means whether that DNA is meaningful, and then DNA makes some RNA, and from that RNA you are getting protein or not. So I explained during my last class that genome size, I explained what is genome, that total amount of information, and gene number and complexity of an organism, they don't correlate with each other, right? Some complex organism like human, they might have less number of protein coding genes, as you can see here, and much simpler organism like worm, that is called here C. elegans, and it has more number of protein coding genes. But what matters is the crosstalk between different pathway, different system, and different genes, and that makes more complexity in our body. So let's see. We will discuss about transcription. So here the total DNA content versus transcribable content. That means like we might have a lot of DNA, but we have to see how much DNA get converted into RNA. Okay, so that's the question. And now you can see here that in case of E. coli, this is E. coli bacteria, right? They have uh, 4.6 into 10 to the power 6 base pair, that their genome size. And in contrast, you can see in human, we have huge number of base pair as our genome. You don't need to remember this number, that is irrelevant. You shouldn't even try. But just I put these two just to have some idea. Now, if you see, protein coding sequences is just, say for example, whatever present here, that many number of nucleotides, right? This is present in every cell in our body. Remember, multiple time I told that we have approximately 10 to the power 13 cell, right? and each of those cells, they contain this amount of nucleotide pairs. This is the total information. But here what I'm trying to say, that out of those information, only out of this sequence, only 1.5% approximately, because in some literature you might get little bit less or more, approximately 1.5% of total DNA content that makes protein, and rest of the things we don't know. Some of those things are currently coming into picture, that how that also helps in our body, or what is their role. But I would say only 1.5% DNA that can code for protein, and that DNA, that 1.5% DNA, when that DNA makes RNA. Just a minute. I think some student actually. So please mute yourself. I think one student. Okay. So now that 1.5% DNA that makes protein. So before making protein, you have to make RNA, right? And from that RNA, you will get protein. So when you are getting protein from some RNA, and that RNA is called mRNA, one new term I didn't discuss before. mRNA. M stands for messenger here. Messenger means it contains message how to make the protein, or what will be the protein, what kind of protein will be formed, like that message is present. So that's why we are telling it as messenger RNA, mRNA. So DNA to mRNA, how it will be formed, the process is called transcription, and mRNA to protein, the process is called translation. I will discuss both of them now.
Sir,
Hmm.
in the previous few slides, we just had DNA to RNA process described by transcription. So only the DNA to the mRNA we are calling transcription, not the other RNA.
No. So just if I go like 2 minutes more, you will get it. So DNA to mRNA and two more defined types of RNA present, which are very important. All those RNA formation is called transcription. But honestly, if you go through any textbook, they will only concentrate on the formation of mRNA, because from mRNA you are getting protein. That's the reason. But in order to get even protein, two more or two different types of RNA is also required, and those are absolutely required. Otherwise, you cannot get protein. I will just discuss now within a couple of minutes. Okay, Tanmay?
Yes, sir.
Yeah. So now, apart from this protein coding regions, whatever like Tanmay just asked, what I would like to say that DNA can be transcribed into two more defined types of RNA, that is called, like again, so this is DNA, and you can get ribosomal RNA, or it is called rRNA. DNA, it can make transfer RNA or tRNA. You can see that R stands for ribosomal and T stands for transfer. So now you will have like total three defined RNA: mRNA, rRNA, and tRNA. So I will tell you to remember these three terms. As I told you that you don't need to remember this number, but these three terms are required, because I will discuss that again and again. But like I'm explaining that's why that RNA, all of you know what is RNA. Now this is messenger RNA, this is ribosomal RNA, and this is transfer RNA. Only 1.5% of total DNA that make mRNA. And what about rRNA and tRNA? I would say their percentages are even less. They make some kind of specific product. So ribosomal RNA that helps to make ribosome. Do you know what is the function of ribosome?
Protein synthesis.
Protein
Yeah.
synthesis.
To make proteins.
Or translation. That's why just few minutes back I told that those RNA are required, or those are defined types of RNA, they are also required for protein synthesis. So RNA, they are components of ribosome, and without ribosome you cannot make protein inside your body. Similarly, tRNA, they are also important. They bring particular amino acid in the translation site. So you might not get it now, but when I will discuss about translation, then I will discuss about its role. So that means here I am just writing help in translation. So that means ribosome and this tRNA, they makes the machinery for translation and the information present in mRNA or messenger RNA. That's all. Most of the DNA sequence are not transcribed in our body, right? But in contrast to human, in case of E. coli, you will see that most of the DNA, they are actually making RNA, and they, I would say, from that RNA you are getting protein. So as a result of that, what you can say, in our case, we have lot of DNA that is called junk DNA. So you will get it in Google or any textbook. That's common term, that junk DNA. That means DNA which are not useful, not required in our body. So then the question is coming here now, that then why this DNA is present? What is the advantage of having this large amount of DNA? Because now if I ask you that tell me whether it is advantage or disadvantage. So if you go by logic, I would say most of you will say that this is a disadvantage, because if you have lot of information, as I told you, like it's kind of huge number of nucleotides present. Now, during each round of cell division, you have to replicate. That means whatever the machinery is present in our cell, they need to replicate the total information, right, in order to divide the cell. And that is, I would say, the wastage of huge amount of time and the energy. Energy means the cellular energy. Then what's the advantage? Then I would say E. coli is more clever, that only harboring those genes which are important, so that will make product, and from product it will get it, like whatever its like required function. So what do you think? Is it advantageous?
Sir,
Yeah.
can I answer this one?
Yeah, try.
Sir, one advantage could be like during replication or during these steps, mutations might occur. So if the mutations are occurring on the important parts of the genetic material, which are going to be included.
Yeah, very good. It's absolutely nice answer, I would say. So just I'm explaining because I stop in the middle to you. So what Som is saying, that is the primary reason, because that's why most of the time you will see that in evolved organism, complex organism, we have huge amount of DNA, and that is non-functional junk DNA. So the idea is, say, like E. coli, most of the bacteria, they survive for like few minutes. That means within minute they will get double, right? Like as I told you, like E. coli within 20 minutes it will get double, and after that again it will get double. As a result of that, in a population of E. coli, if one E. coli is wrong, some of its DNA it contains wrong information. It's kind of very kind of that is called mutation. Actually, whatever Soham told, that problematic information, that E. coli will not survive; it will die. But we cannot do that, right? We stay for a long time, and we have huge number of cells, and we have to be, good enough about respect to our information. So as a result of that, if we keep lot of DNA and the precious DNA or the information, those are just hidden. Like we have only 1.5% of DNA that encodes protein. So as a result of that, always we are challenged with some mutagen. Mutagen means something which can cause mutation in your DNA. Mutation means some changes in your nucleotide sequence, right? So, for example, sometimes we eat lot of junk food, and that contain some bad quality color also. So that also causes some problem. Sometimes radiation, and so many things are actually there. So as a result of that, if we have like DNA, less amount of DNA and 100% are required, without that you cannot function, then it will be very much problematic. If you like some of your DNA, they get mutated or some kind of problem happen. So that's the reason why like generally higher organism they contain lot of information, but only few information they are actually required for the function. And not only that, here I would like to stress one thing, that that information also present in 2 times in every cell. Again, I am telling you, whatever the important information present in our DNA, in gene, that is also present 2 times in each cell. Can you tell me why 2 times I'm telling? Any link?
23 pairs of DNA molecules, I
Yeah,
think.
yeah.
So one
Because
is recessive, one is dominating like this.
exactly, because we have 46 DNA, 23 we have obtained from my, say for example, I obtained from my mother and 23 from my father. So apart from that X, Y, that sex chromosome, the all other informations are present in twice in every cell, and that is again beneficial. If for some reason one gene is getting mutated or some problem happen, but still other will support, the 50% of your product will be there. So those are the complicated thing, and that helps. So in E. coli, all those information present just one time, not twice. Okay, so they have just haploid information only one time, those information present. So now that's the reason. And now I will just discuss briefly what is the mechanism of transcription. So in between, just I put this slide. What is a machine? If I ask you, maybe all of you want to you will try to answer the question. But before I would say, even I just put some answer from Cambridge Dictionary. So what is a machine? A piece of equipment with several moving parts that uses power to do a particular type of work, right? This is some kind of classical definition. I would say those are like real or big world machine. But if you see like molecular machine or biological machine, like DNA polymerase, RNA polymerase, ribosome, they also satisfy all those all those like key points mentioned in this definition. Like they also have moving parts in this enzyme or in this protein, because they are not like rigid body. While they are functioning, they can also move, some of the portion moving, and that's why they can catalyze the reaction that uses power. All these enzymes or machinery, like ribosome is not, you cannot say really an enzyme, but they use energy in the form of some ATP or GTP, like some molecules, but they use power to do a particular type of work. So for example, DNA polymerase, it polymerizes DNA bases, so it synthesizes DNA. Similarly, ribosome that helps in translation, or it continue translation. So that way they continue specific work. So all those things are kind of biological machine or macromolecular machine. So now, again, I will go slowly, because in this slide you will get the idea of transcription. That means you have to make RNA out of your DNA sequence, right? So in this case, you can see here, this is the DNA or double-stranded DNA, and you can extend this DNA, as you wish. It's kind of long DNA. I told you that some of the DNA are very long in our body, right? So I'm just showing here little part of it. And one DNA molecule that can contain multiple genes. So for example, one gene is mentioned here, gene 1, and maybe another gene present here, and that is gene 2. But our goal is to understand how this gene can be transcribed into RNA. So what happened? First, you have to understand that how, like I told you, that's only 1.5% of like DNA that can make or that form mRNA or protein coding, like mRNA. But the thing is how our system will understand that where we need to start transcription, because all are double-stranded DNA, right? So you have to find out the portion. Yeah, so we have to find out the sequence where you have the information and that need to be transcribed. So in order to get that, some kind of machinery present here, you can see here, that is called promoter. Promoter is present just upstream of your gene. Upstream means just above your gene, like little bit away from your gene, and this promoter actually invites one enzyme that is called RNA polymerase. So again, I'm telling you that this promoter means nothing, some kind of DNA sequences, okay, some kind of specific DNA sequences. That means specific order of nucleotides present in this region of DNA, and that attracts this enzyme called RNA polymerase. So as a result of that, RNA polymerase will bind here in the promoter region, right? This is your RNA polymerase. And during replication, I told you that there are a lot of things like helicase, guidase, many things are present there. But in this case, RNA polymerase, it has the capability to unwind the DNA, right? Just one enzyme is enough. But you have to remember that this enzyme is not transcribing the whole DNA, like whole long DNA, only a small portion, which is called as gene here. That
So,
RNA polymerase,
which
yeah,
means that only the gene one portion will be unzipped, not the rest. Like in case of DNA replication, we had to unzip the entire DNA. We had to unwind the entire DNA molecule, but here only the important
gene.
genes which will be transcribed. Only that part is.
Yeah, right. Only gene one. Say, for example, you have like in one DNA, you have gene 1, 2, 3, 4, 5, 6. Just for example, say, for example, at some point of time, you need to make protein from gene 1 only, but not from other genes. That is based on your requirement. I will discuss today how it can be decided that which gene will be turned on and what gene will be turned off. I will discuss that today. But this RNA polymerase will take care of that portion only, that what is required during that point of time. And as a result of that, it will make some kind of bubble. Bubble means that two DNA will be displaced, and RNA polymerase itself, it has properties. So as a result of that, since it's a small segment, so as a result of that, it will not like pose that kind of constraints. In case of DNA like replication, it has huge problem, but in this case it should be fine. RNA polymerase can take care of it, and it will make the RNA. And also the process is much slower than replication. Replication is very fast, like huge fast, I would say. I forgot exact number, but transcription is very slow process. I would say in E. coli, the rate is around 45 to 50 nucleotides per second. This is actually a slow process. And now, if I go back here, so now, since you have like single strand available here, and this is because of this RNA polymerase, and now what will happen? The RNA synthesis will take place. Here you can see, again I'm going back here. So here you have the RNA polymerase, and it will move in this direction, and while it is moving, it will synthesize the newly, I would say, the nascent RNA. Here you can see this is the RNA that being synthesized, okay, and this is should be always 5' to 3' direction, right? Here you can see 5', and it is going into 3' direction. And RNA is single stranded. You don't need to be, concentrated here on the upper strand here, whatever strand mentioned here. So as a result of this, so whatever sequence present here in this strand, you will you will make your RNA based on the sequence present in this strand. I will discuss in more details in the next slide. This is just the introduction, and this is called elongation. That means the synthesis part. And then finally, some information present here, some sequence present here that will help to remove the RNA polymerase from the DNA. As a result of that, only you will get the product of gene 1, that is nascent mRNA, which is coming from gene 1. So that is your nascent mRNA. Then RNA polymerase will go away, and it can catalyze another round of transcription. So now, like 3 key points.
So can you please repeat the nascent mRNA portion?
Yeah, I will discuss, because like if you see my point here, like coming 3 slides will be dedicated to discuss this concept. So key point to be discussed in the following 3 slides is promoter. So here, what is promoter? The second is RNA polymerase, and the third one is RNA synthesis, like nascent mRNA. Nascent means just synthesized. Nascent is just English term, so that means just synthesized or just formed. That is called nascent. mRNA, because this is protein coding genes we are discussing, that's why mRNA. If it is not protein coding genes, then it could be RNA or tRNA. But let's concentrate on these 3 points. Then I hope you will get everything clear. If not, then stop me and ask me. Now concentrate on promoter. So promoter is just like a pointer that points to the location of the information or gene to be copied into mRNA. So pointer, this term, even I believe you have studied in your, now you are in first year actually student, because we used to discuss this even before second and third year student. So for example, you are playing some DVD player or CD player, okay, and you have like 50 different types of songs, those are present in that CD or DVD. But you can select whatever you like based on some pointer mechanism, right? So you can point that particular information so that you can listen that music or that song, whatever you like. So similarly, like promoter, you can, the analogy is just like pointer, that promoter actually decide which gene at what point of time it will be transcribed. So their role is to point the RNA polymerase or to show RNA polymerase, yes, you have to start from here. That's the job of promoter. So now you can see that if these two are the DNA strand, the 5' to 3' and 3' to 5'. I am going very slowly, carefully listen every word and sentences. So those two are the 2 different strands of DNA. So as a result of that, DNA runs in anti-parallel directions, so 5' to 3' and 3' to 5'. This is important. That's why I'm telling you, because when we are talking about RNA synthesis, this direction is also important. And now, if you see here information, so here if you have like A, T, T, G, C, this kind of information, and you should have just complementary information in the other strand. Like, again, you shouldn't remember this information, but what I'm trying to say that if this is the starting point of a gene, right? This is a gene here, like this is started here. This is the starting point of gene. The first nucleoside is denoted by +1 site, okay? And then this should be +2, this should be +3. That way we mark the nucleotide of a gene. And then what will be nucleotides present on upstream? Upstream means before the gene. So for example, something present here, like before A. So that should be -1, then -2, -3, something like that. There is no 0 in this case, like +1, +2, +3, and before the gene, you have -1, -2, -3, something like that. But that is how we actually label those nucleotides. But now, remember, when the RNA is being synthesized, then what will be the RNA sequence from this gene? I'm just giving you the sequence here. See, this is 3' to 5' DNA. So it will be used as template, and you will make. So template means this. Here you have to be very much clear. So otherwise everything will be problem. So if this is your DNA, this is 5 prime, this is 3 prime, 3 prime, 5 prime. RNA polymerase is synthesizing your RNA. So what will happen? You cannot. Can you tell me, please? Okay. So if you see that RNA cannot be synthesized from 3' to 5' direction, any nucleic acid, right? So as a result of that, you have to start synthesis from here in this direction, right? So this is 5' and this is 3'. Then how it will be synthesized? Say for example, RNA polymerase will also add complementary nucleotide. That means if you have just for example A, G, C in this strand, this is called template strand of DNA. Template strand. Then what information you will get here in the RNA? This part go very carefully. If you have A, then it should be what in the RNA? Can you tell me?
TCG. TCG.
Not T, it should be U because this is RNA, right?
Yeah.
Yeah, this is fine. You can tell T, but like by mistake, but it should be U. It should be U, and then it should be C, and then it should be G, and it will be continue like this because whatever information present here in the template, just complementary sequence will be coded here. So now, say for example, you have here another T here in the template. So you will have A here. So that way it will be continued. So that's why this strand, the lower strand in this case, whatever I'm drawing here, that is called template because on the basis of that, this RNA is getting synthesized. Now, can you tell me that the sequence of RNA actually will be similar to which strand of DNA, the lower strand or the top strand?
Upper strand.
Upper strand.
Right.
Very good. Yeah, because if you have TAGC in the template strand, the corresponding sequence in the upper strand should be A, T, C, G. Because see, that's why this is double-stranded DNA. So here you have TAGC, so here it should be ATCG. They are complementary. So as a result of that, whatever the RNA you are making out of this template strand, their sequence will be exactly similar to the upper strand here. Apart from you will have uracil or U instead of T. Otherwise, everything will be same. That is the reason why we say that the lower strand in this case is template strand and the upper strand is called coding strand. I spent a lot of time just to explain this, why it is called coding strand, it is called template strand. Otherwise, everything will be, you will forget everything. So even when I studied, I used to forget every time. Then slowly, teaching several times, and then I got this idea that, at least how I can explain that. So this is called coding strand or sense strand. That means sense means the sense is present here. The information is present here. The code is present here. That's why this strand is called coding strand. But how you can make the RNA? So you have to use the template strand and you have to synthesize just complementary sequence which is present in your template strand in the RNA, and then you will get the information which is exactly similar to the coding strand, and then that RNA will be utilized as mRNA, or that will make the protein, because whatever information present here, I didn't discuss the mechanism yet, but on the basis of that you will get your protein. So let's see whatever present in the slide. So here this is coding strand and this is template strand I just discussed. And now as I told you that we are discussing about promoter. So I already discussed what is +1 -1 side. So in prokaryotic system, prokaryotic means like for example bacteria. Okay, bacteria. So prokaryotic stands for, so this is Latin word, you can forget it, but I'm telling you so that you can remember. So but if you if you forget, nothing will happen. Karyotic, it is coming from a word called karyon. In Latin, that is called nucleus. Okay, so prokaryotic means before nucleus. Bacteria, they don't have nucleus. They have just DNA inside bacteria. But eukaryotic means after nucleus, like we have nucleus and DNA is present here. So in this context, please forget it. Again, in a defined class, you will know about this. But in prokaryotic organism, for example, in bacteria, we have some kind of specific sequence, like you can see here, TATAAT, that is called promoter, at around -10 position. And that invites RNA polymerase. That means RNA polymerase can recognize, okay, RNA polymerase can recognize this sequence, and then when RNA polymerase will bind here, it will make a single stranded DNA just front of it, and it will synthesize the RNA here, like the mechanism I told you here. Similarly, in eukaryotic system, little bit different and more complexity is there. We have
Sir,
around in minus.
Sir, one doubt, sir.
Hmm.
The like that TAAT is
Hmm.
present at -10. So exactly how, if exactly after that the RNA sequence starts getting created, so in between there will be some sequence that might not be useful to the RNA. So what about that?
Yeah, very good point. Actually, that is kind of more mechanistic details, but intentionally, I was ignoring this part, but you are right.
sir, I have one more doubt.
Yeah.
Sir, here the given like sequence is only composed of TNA. So like, is it something coincidental that has happened here, or is it always TNA?
Okay, okay. I understand. So there is some reason actually why it is TA. I'm coming into that. Both the question I'm trying to address it. Okay. The thing here, so in this case, the first question, whatever asked, that in between this +1 side and this side, some nucleotide sequences are there. But the thing is, in the initial part of transcription, like some, that is called abortive transcription. That means some transcription start here. Like if RNA polymerase sit here, RNA polymerase is little bit bulky, like little bit bigger enzyme. If it sit here, then it can start transcription almost 10 nucleotide away from it, because it's a kind of, like if RNA polymerase bind here, maybe its active site is little bit away, so that it can start transcription from here. So that's why, so for example, if RNA polymerase bind here in the +1 site, then it would have start the transcription little bit later, and that way you will actually lose some of your information. So that way it has been evolved, right? And now, in order to answer the second question, I would say that TATA, this kind of sequence, I most likely this is like some kind of like coincidence in this case, because when RNA polymerase bind here, so this portion, like primarily not not getting separated. If I would say, if this portion need to be separated at first, then I would say there are some reason, because if you have multiple TA, then you need less energy, because they have just double bond. But as far as I know, this portion is just for recognition, not to like initiation of the separation of these two strands. So as a result of that, most likely this is just some kind of coincidence. And also I would like to tell you that another region present here, just I didn't mention here, just to keep it as simple as possible. So in this region, like this is also promoter. In eukaryotic case, sometimes we have multiple promoter. So here, like most of the sequence are G series. So the second question, whatever like you asked, I will see little bit about it, and I will address during next class also, that apart from this, whatever I am telling coincidence, whether the initial, like whatever the sequence it is being recognized by RNA polymerase, whether that needs to be separated or melted at the beginning. If that is so, then it is advantageous because you have just TTA, this kind of sequence. So you need less energy.
Okay, sir, got it.
Yeah.
Yes, sir.
So
I
I will
also.
check it. But this is good question. I didn't think about it before, but this is good question. So I will check it, and like, you know, if whatever I told, if this is correct, then it should be fine. If I get some, you know, better information, I will you know, address that in my next class. Sure. So let me go through the next point now. The RNA polymerase. I told you that three defined points we will discuss. The first one, promoter, I discussed. Then the RNA polymerase, the main machine or the enzyme here. This is enzyme. So enzyme means you have to remember the RNA polymerase itself is a protein, right? Itself is a protein. So I will come into that discussion or debate later, because in order to make protein, you need multiple protein also. That debate I will discuss a little bit later. So now, subunits of RNA polymerase. That RNA polymerase, just one enzyme is required to, you know, for the transcription. You don't need several enzymes, just like DNA replication. Here, one is enough. But in this RNA polymerase, you have multiple subunit. That means, you know, multiple different things they assemble together and they make the RNA polymerase. Okay, these are the subunit. Two alpha units, two beta, beta prime, and sigma. I should mention you here. You might not get the link little bit because we didn't discuss protein yet. Because I have to discuss all those things in order to explain you protein in details. So this alpha, beta, sigma, those are different small protein. All of them, they assemble together and they make the enzyme called RNA polymerase that catalyze this transcription reaction, or that can synthesize the RNA. So as a result of that, if I again like go back here, like this is your gene here, and it needs to be transcribed, and this is your promoter, right? This is your promoter. So as a result of that, this promoter will be recognized by RNA polymerase. I told you before. But if you see little bit in more details, that this promoter is recognized just by this sigma subunit, just sigma. That means during the starting point, during the initiation of transcription, all those subunits, they are not staying together. The sigma stays alone. So sigma is a subunit, or it's a protein. It has the capability to recognize that sequence TA, whatever I mentioned there. You don't need to remember the sequence, but I would like to mention that sigma can recognize this promoter sequence. What we are talking about, this is true in case of E. coli or bacteria. Most of the explanation we concentrate ourselves in E. coli because much, I would say much simple. But in eukaryotic system, the overall concept is same, but some more complexities are there. And then the job of sigma is, like, although we have very long DNA, but sigma will find out that where the transcription need to be started. That means that it will just find out the promoter side. And then what will happen? The rest of the like subunits, like alpha, beta prime, they will join together. So this is the complete RNA polymerase. Okay. And as I told you, they have moving parts. Yes, this beta prime, they are different protein and they can move little bit in order to catalyze the reaction. And now if you see, before like actual transcription, that means when you will make the RNA, this sigma needs to fall apart. This sigma needs to go away from the complex. The job of the sigma is to, just locate the region, and then when the sigma will fall apart, then the RNA polymerase, that contain alpha, that means 2 alpha beta beta prime, they will move this direction and synthesize the RNA. So can any one of you guess that why sigma needs to be, get rid of from this system? I didn't tell you the answer, but some imaginary answer that why this sigma needs to be, get rid of from this complex.
So it itself binds, maybe binding to the some of the nucleotides, which may block the others.
Another mRNA synthesis.
Yeah, so there are two things you can say. One is you could say that like its job is done because its job is to locate the promoter. It is done. So if it fall apart, it can be recycled and it can be used in another transcription, like for another gene. Because I didn't tell you yet that for every gene, whatever genes present in E. coli, just one type of RNA polymerase present. That means only one type of sigma also present. In our body, we have 3 different types of RNA polymerase. I am not going into that complexity, but we have 3 different types of RNA polymerase. Like one RNA polymerase make mRNA, the other RNA polymerase make RNA, and the third one on tRNA, right? But in prokaryotic system, or all RNA polymerase, they are same and they make all sort of RNA. But in this case, like the first thing is sigma can be recycled, and the other thing is sigma actually locate the region. That means sigma has the high affinity, the high binding with the promoter sequence, right? So as a result of that, if it like binds to promoter sequence tightly here, like this sigma, like if it binds to promoter sequence tightly, then the whole complex might not move in this direction, right? So as a result of that, there is some mechanism so that displace the sigma, so that now the location is fixed from where you have to start the transcription. Then the rest of the portion, that is called core enzyme, right, which has the catalytic power. So it can move like freely towards this direction, and it can synthesize the RNA. So those are the two reasons why sigma needs to be get rid of from this complex, and then it will make RNA. Now I will discuss 2 points here, like in comparison to DNA replication. The first thing is the RNA polymerase is completely processive. What is the meaning of this processive thing? That means our transcript is synthesized from start to end by a single RNA polymerase molecule. Say, if you have one DNA molecule, I am not making the helix here, like one DNA molecule. During transcription, it is getting separated like this. Remember it, that only one RNA polymerase, it start the transcription here. This RNA polymerase will complete the synthesis of RNA throughout the gene. You don't need to recruit another RNA polymerase. So that's why we are selling that completely processing. But what happened in case of DNA polymerase? As you can remember that in case of lagging strand synthesis, you need to recruit multiple DNA polymerase, right? Otherwise, you cannot synthesize the lagging strand. But in case of RNA polymerase, whatever the RNA synthesis started, it should continue. For some reason, for some accidental case, if the RNA polymerase get detached, then again another RNA polymerase cannot find the location. So that RNA will be destroyed. It will not be synthesized anymore. Okay, so this process is completely processive. One RNA polymerase start the process, and that RNA polymerase needs to finish the job. That's all. Okay.
Excuse me, sir.
Yes.
Sir, in the lagging strand, so all the parts of strand synthesize simultaneously or one by one? You said
In the
that
lagging strand, one by one. Otherwise, the rest of the portion not yet open. When the replication fork is moving, if you see that the replication fork is moving, the other portions are not yet available. Is it clear, Rajas?
no, sir, but you said that many DNA polymerase are needed. So all of the means which portion is open. So all of the strands need to be synthesized simultaneously, no? So to means take all the DNA polymerase, means use
So-
all the DNA polymerase.
So just a minute, because I know that this kind of confusion might come, but let me little bit introduce. So during DNA replication, what happened? If this is 5 prime, 3 prime, 3 prime, 5 prime. One DNA polymerase here, it is synthesizing the DNA continuously. And as a result of that, this DNA can be synthesized continuously by one DNA polymerase, because like after some time, what you will get? You will get the fork here, right? You will get the fork here because the fork is moving in this direction. And here you will get the double stranded DNA because it is already synthesized, right? Right. So now this DNA polymerase can synthesize the DNA like continuously. But here I told you that DNA will be synthesized in short segment by multiple DNA polymerase. Because why multiple DNA polymerase? Because this DNA polymerase is done, then this portion will be opened, again it will be synthesized. So as a result of that, for lagging strand synthesis, you need multiple DNA polymerase. Is it clear?
Yes, sir. So one DNA means the previous DNA polymerase cannot be utilized for the further strands.
Yeah,
Means
yeah,
parts.
yeah,
Okay, sir.
exactly. So that's why. So in this case, but in case of RNA polymerase, just one RNA polymerase will start and end the process. The next point is very important because you didn't ask even, because since like many of you actually asking many questions, but this is important one, that RNA polymerase, then you could have asked me that sir, why primer is not required here, because initially you told that for synthesis of nucleic acid you need a primer, but that was the property of DNA polymerase. Okay, that was the property of DNA polymerase. It cannot start the reaction by itself. But fortunately, because have you seen that in case of primase, that is some kind of RNA polymerase. That primerase is adding RNA nucleotides, right? So similarly, here you are making RNA. So RNA polymerase has the ability to add the first nucleotide, right? So it can initiate the process. If you can start that, if you can ask me that why RNA polymerase can initiate, how it is getting the 3' OH, that is like clearly known. But I'm not discussing, because mostly for our master student we discuss at that level that why RNA polymerase can start or initiate the process. But at this stage, I would just like to mention you that primase that make RNA primer. RNA polymerase also the similar thing, that they make RNA, right? Those enzymes, they have capability to start the process. So as a result of that, what you are getting here, that RNA polymerase can initiate the synthesis of RNA de novo. De novo means here that no primer is required. So that's why you can see that no
Can
primer.
you repeat the reason, like why is no primary required in case of RNA?
I didn't tell you the answer actually. I just told that primers are required because DNA polymerase cannot start the process. So DNA polymerase needs some pre-existing 3' OH group of some nucleotides so that it can start the reaction. But RNA polymerase can like start the process by itself. So the reason is very complicated. That's why I'm not telling you. Just within like 30 seconds, I would like to tell you, but like as I am telling you, that don't go for it because it will be more complicated. Because in RNA polymerase, like this is protein, and proteins are made up of amino acid, right? So one of the amino acid, they are in their side chain, that amino acid called tyrosine, and in their side chain it has one hydroxyl group OH, and that is actually present in the like active side or the catalytic power. So that OH initiate this nucleophilic attack in case of RNA polymerase. Again, I am telling you, you don't need to go in that level of details. But since you asked, I cannot completely ignore also. But the thing is, RNA polymerase, they can initiate the process because that 3' OH, which can like which can actually help in synthesis of DNA, like because of the nucleophilic attack. But in this case, RNA polymerase, it contains OH group in its protein sequence itself, and that is actually that can start the process. This is, I would say, very advanced level of research also, like, not even you will not get it in like every textbook, but at this level, I would like to tell you, just forget about it. Just remember that RNA polymerase can start the process without having primer, right? That's all.
Okay, sir.
Yeah. So now, for this reason, like how RNA polymerase like overcame transcription, right? So how actually it helps. So for that reason, sorry, I already wrote many things here. So for that reason, like Roger, he got Nobel Prize. So he is Roger Kornberg. In 2006, in chemistry, he got the Nobel Prize. Okay, so and here you can see his father, Arthur Kornberg. So can you remember who is Kornberg, Arthur Kornberg? I like discussed in during last class, I believe. So he discovered the replication, his father, and son he discovered like contributed like huge in transcription. So you can see this looks like almost their kind of family trend, that two important, like, replication, transcription, translation. Those three are the central dogma or central idea of like modern biology. So one is contributed mostly by Arthur Kornberg, and the next step, the transcription, like mostly contributed by Roger Kornberg. And both of them, they got Nobel Prize for their contribution in chemistry in different time, right? So it's kind of huge for them. Now the process of RNA synthesis, I already mentioned, nothing new here. So here you can see this is the single strand, like, this double stranded DNA, this is the template strand, coding strand, all those things I told, and the overall mechanism is same. So it will add the, the next nucleotide in its 3' position, right? So as a result of that, whatever the growing RNA chain is present here, and as a result of that, in the 3' OH group free here, so it will catalyze the reaction here. So the next nucleotide, the N+1, so that will be added here. So as a result of that, you will get N+1 residue. So that way, the RNA will be synthesized in this direction. And this is about RNA synthesis. And then, yeah.
Sir, I have a doubt that what was that TATA thing?
Can you tell me one more?
Sir, TATA box thing.
So where you saw in this slide or before?
Sir,
In
2 or
case of
3 slides back.
Yeah,
eukaryotic,
I know that TATA box, right? So I believe you have seen here. So
Yes, sir. These two things.
Tata box. So this is
TATA.
just their common name actually. The Tata box, this is just a promoter. That in textbook you will get in that name, the Tata box, because they have this TATA sequence. That's why, nothing more than that. Okay. So this is called Tata box. And
So it
now,
is always TATA.
no, so this is so that's why I would say this is mostly T and A rich, but this is not like 100% conserved, right? This is mostly TA rich, but not 100% conserved. Okay, so that can be recognized by RNA polymerase. That is how it works. So now, in this case, I just forgot. I thought something to discuss. Oh, okay. So now, then the RNA is getting synthesized, right? Then what will happen? The like the next point is the transcription stop. How it will be stopped? So that previously we included it in our slide, but recently we removed it because of online classes, because it is difficult to explain in online mode actually. So I would like to tell you, when the gene is done, then you have some signal sequence, and that signal sequence actually help to displace or remove the RNA polymerase, so that RNA polymerase cannot move forward and the transcription stop there, and you will get the nascent RNA. Nascent means newly synthesized RNA. So now please take 4 to 5 minutes off. I am also taking break, and then I will come back and discuss the next part, that how like we will understand that which portion of DNA or gene needs to be transcribed at certain point of time. That is the control mechanism. That how it will be decided that what gene will be transcribed at certain point of time. That what is the mechanism of turn on and turn off, right? That we will discuss after 5 minutes. So please take break for 5 minutes.